Welcome you all to this live practice session for the Applied Environmental Microbiology course. We are in the week 11. So we just have one more week left. So if you have any doubts, you can ask now or maybe in the next class also. If you're planning to take the exam, before the exam, you may ask it during the session so that if I'm able to, I'll help you out with that. So week 11 course content focused on two topics, which was epidemiology and biosensors. I just have few practice questions related to these topics. Let's get started now. So first question is, what is a sensor? This doesn't have any exact one right answer or wrong answer. I just wanted to get a sense of what you think is a sensor is. So we'll start from the basics here. This can have multiple perspectives, so we'll just get to know each other's perspective. Very simple. All of you can come up with just few words or one line answers. All of us know as for enough time now. I guess all the participants who are here are there for. I know few who are there in all the 10 weeks or maybe more than 5 weeks for now. So all of you can answer. We'll get to know. Okay. Two people have answered. Any more answers? I guess there are two more in the meet. So even if you feel like it's a repetition, you just can give your answer. Not an issue. Okay, as far as this question is concerned, I haven't given any answer in my slides. I just wanted to know what you think. So basically, a sensor in a very raw sense means something that senses. As the name suggests, a sensor can be a device or a system that basically senses something. Okay, that is why it's called a sensor. We use sensor in multiple places. Like a basis of a sensor comes from the domain of electronics engineering or electrical engineering, maybe something of that origin. So it senses some changes, as you have rightly mentioned, changes in the environment or some defects, as we can tell them in some cases. Not in all cases they are defects, but in some cases they are defects, and in most cases they just sense the change, what should be there, what is actually there. So the change between these two parameters is being sensed by some instrument using maybe a reaction. In our case, in biosensors, we mostly see some biochemical reactions driving this, right? So we have some reactions or some form of detection of this, and that is finally given as an output for us, which is quantifiable. We also have qualitative, uh, but we also go for quantitative analysis using a sensor. Sensor is widely used in various fields, okay, various places. Can you just give few examples of sensors? Not only related to biology or environment, in general, you can give. Just let me know if the chat box is accessible to all of you. There is no issue in that, right? If that is accessible, just give me a thumbs up. I guess reactions are always on, so okay, great. Before understanding about any particular sensor, you have to know what a sensor means, because that is used varied in varied fields. Whatever you are going to be working on in the future, you can use sensors. So any examples of sensor? It can be a biosensor, it can be a non-biosensor, doesn't matter. Whatever you have studied in the week 11 content, even that you can tell. pH meter sensors, great. Right, that's a very great answer. Skin is a great sensor. So this was what the discussion had to be oriented to. As I told you, a sensor is something that senses changes, right? Senses a difference in the environment. So the very basic sensors that all of us know is all the sensory organs that we have. So we are able to detect senses, right? We have eyes, we have ears, so we have skin, as very rightly mentioned. So what do we do with our sensory organs? We just sense something. We just sense the difference in the light rays, the wavelength that's emitted that's reflected or that's absorbed, all this play a role. But at the end of the day, what we perceive is an image of what's in front of us. So eye is one of the sensors. Skin is a very good sensor of temperature changes, right? We can sense when it is cold, we can sense when it is hot, and we respond according to it. So uh, before getting into biosensors or other technical details of how to build it, what's the use of it, we just have to have a very basic understanding of what a sensor is. I guess it's clear now. So with with this basic understanding, maybe we'll go to the next question. So if you have any more thing to add, or if you want to ask anything, you can just put it in the chat box. We'll look into it. So with this little introduction about sensor, we have seen that biosensors uses biochemical reactions to detect the target. So this is what I, okay, so you can try this question now. Okay, so I think from the four responses, three are right. So electroactive substances use electrode, light photon counter, mass change is piezoelectric effect and heat is a thermistor. So first coming to the piezoelectric thing. What is a piezoelectric effect? How does that sensor work? Any idea? Okay, receptor interacts with the analyte and undergo change, and the mass added gives change in frequency, and that is amplified. Yeah, you're right. So to start from the basic, piezoelectric effect is something which is very simple. We have learnt in our higher secondary that it refers when we are producing stress over a material, it generates some voltage, and inverse thing is also possible. Like when it is mechanically stressed, it produces voltage. When we produce voltage, creating mechanical stress over a mass is possible. And this usually happens with piezoelectric material. Uh, Generally, they are crystals, anisotropic crystals. So what happens is when we are adding, as you told, when we are adding this mass, uh, the signal that receives has a different the oscillations or the frequency in that chemical is different, and the signal that we receive will show detection. So that is how a piezoelectric sensor works. Basically, and just have the piezoelectric effect in mind. So imagine individual molecules. So when we add mass to it, the frequency and the oscillations are going to change, and this change in oscillation um, will be detected. The change in oscillation can be brought about by stress or by voltage. So like the piezoelectric effect and the inverse piezoelectric effect, either of it can work for the construction of the sensors. So there are few pH sensors which use this piezoelectric effect, and we can also have antigen antibody interaction kind of a sensors can be developed using this piezoelectric effect. So it has a wide advantage, and there are also many research works going on this. Okay, and then coming to the thermistor, what is a thermistor? Also, let me know if whatever I'm explaining is not clear. I'll repeat it. Okay, so let's talk about thermistor now. We all know thermistor is going to sense heat changes. Yeah, thermal basically. How it does that in piezoelectricity? We saw that mechanical stress applied over a material induces voltage, or voltage applied to a material induces mechanical stress. So this is going to cause a difference in oscillations in the crystal, and that can be detected. Similarly, in case of thermistor, what are we detecting and how are we detecting? Okay, they maintain a specific temperature, and other person is telling it's a thermostat. So, right. Exactly. So, whatever you have answered to put together is what you have to know about a thermistor. So, generally, the principle of thermistor is that when the temperature changes, the resistance changes, and this resistor is detected. So, this kind of sensors, basically, what we do is we use a ohmmeter or some device, or the device that we use to measure the resistance, and this change in resistance is being transduced as the signal that we want as output from the sensor. Okay, and as you have rightly mentioned, thermistors can sense, and on top of that we can also maintains same temperature based on the resistance change. You can add some more modifications to it where they resist the change in temperature. If they are going to sense some change, they are going to resist that change, and in that case we call it as a thermostat where the temperature is maintained. Okay, and the next one is an electrode. As you all know, electroactive substances are substances which can either accept or uh, donate electrons. So this electroactivity, I guess, in it works with many things around, not only with chemicals or stuff. Even in biology, we have this electroactive substances. So we have these electrodes, basically anode and cathode, one of which can donate electrons and other can accept electrons. So this reaction, electrochemical reaction, basically gives us the information that we want from the sensor. Any change due to electrochemical reactions where transfer of electrons or redox reactions are involved, we use this electrodes as sensors. And light, as we all know, are energy packets and are made of photons. So we use photon counters to detect the change in light intensity, basically. So when the intensity changes, the photon counts also change. So the light intensity changes as the result of some change in the analyte that we have. So in that case, the light changes directly proportional to the change in analyte. So hence we detect it using a photon counter, and we interpret the results in this. I guess the principle of all this and why we use them is clear now. So with that, we'll go to the next question. Which of the following defines the basic characteristics of sensor? I'll just wait for a couple of more minutes. We have different answers. We'll also wait, see if someone else has some answer. Okay, so let's discuss what's linearity. Linearity in measurement includes the consistency of measurement. Okay, so our measurement should be consistent, reliable linear range. Yeah, delta between the two readings should be as minimal as possible, right? So our data should be consistent. It's not like we get one result one time and some other result the other time. It should be most consistent as possible. Ideally, we don't get very perfect answers with sensors, with any instrument basically. There can be space for errors, but that should fall within a range. So that we define as this linearity, where we ensure some consistency in the measurement out of all the measurements that we have made in a range, between a range. And what is chemical interference? Right. Chemical present in the solvent or solute should not interfere with the sensor, right? And selectivity, we'll just get a definition of selectivity and we'll discuss all of it together and see what's the right answer. Okay, interference is something other than the analyte, right? Generally, when we do experiments, also we tend to hear this a lot: specificity, selectivity, and in case of measurements, we hear about accuracy, precision, linearity, too many terms, right? So we should be clear about what each of it means, because our sensor development is going to be based on all of these factors only. Okay, very specific is selectivity, right? I'll just wait for a few more seconds to see if someone else wants to answer. It should take all readings and not be selective. Okay. Okay, so with that we'll discuss. So as we have discussed, linearity is consistency. So the consistency should be high. So yes, it is true. So that is one of the characteristics of a sensor. And similarly, the interference should be low. If the interference is low, we can have a very reliable result. So the interference should be low. But in case of selectivity, what we have is, selectivity means the ability to select a specific analyte. So when you are testing for a particular analyte, having a higher selectivity is better. Okay, selectivity means how selective it is in detecting and giving the output of our sample. So in that case, selectivity low is not good, selectivity high is good. And as low the response time is, as fast the detection can be. So response time low is also a very good characteristic. Okay, so except selectivity is low, all of them define the basic characteristics of the sensor. Okay, so I have highlighted that. Only that is not a basic characteristic, other are basic characteristics. Okay, and here you can see the next question. Yes, right, there should not be false positives. again, if you are interested in the experimental part of whatever we are studying, or in any field, you can just have a look about all these terms that look similar. Okay, especially in the field of biology or biotechnology, if you are working, you can look about the specificity, sensitivity, selectivity, and all these play a major role in sensor development. And in case of any experiment, you can check for accuracy, precision, all such kind of words, and the difference between them. And again, to reiterate, from the exam point of view of this course, whatever scope being discussed in the lectures is enough. I am telling extra because I get few comments from the participants that they are interested in doing further research or taking up a career in similar fields. So I'm just letting you know, if you have a broader vision in this field, you you can go and look upon all the details of it. It will be useful for you. Okay, we have one response. We'll wait for a few more responses and we'll discuss. Any more answers? Okay, two people have answered. So should I wait more or can we discuss the answer? Just let me know in the chat box. Okay, please go ahead. So we have two answers, C and A. What is QCM? Any idea? You can put in the chat box. We'll QCM and SERS. You can just give the expansion in the chat box. Maybe if you have known the expansions before, you would have tried answering. Okay. QCM refers to quartz crystal microbalance and SIRS refers to surface enhanced Raman spectroscopy. Okay, both of these are biosensing techniques only, and I think it's also discussed in the lectures. I saw these words, all these terms I saw in the lecture PPTs. Maybe you can go through them carefully once. Okay, so QCM refers to quartz crystal microbalance. Okay, and SIRS is surface enhanced Raman spectroscopy. I guess now you know what the answer is. So SIRS, as Raman spectroscopy is involved in it, we are going to measure about light scattering. So inelastic scattering and SIRS should be a right pair. Similarly, quartz crystal microbalance is again we are using quartz. It's a piezoelectric crystal or an anisotropic crystal. So we'll go for piezoelectric detection there. In the case of impedance spectroscopy, we have EIS, electrochemical impedance spectroscopy itself. So again, that's a right pair. So the answer here is the fluorescence emits shorter wavelength. Fluorescence generally takes up wavelength and emits a longer wavelength with lesser energy. For example, a UV light can be absorbed and a visible light is emitted. So the wavelength is longer, but the energy is lesser. So uh, that's about fluorescence, and that is the wrong pair. Okay. Okay. So I guess it was clear. Only with the expansion part you would have struck. I think that's why I did not get much of responses from you, and that was intentional. So just go through the PPT once. Just try to understand what expansions or abbreviations we use for what. I think it also had about SPR, which is surface plasmon resonance, and about SPM, which is scanning probe microscopy. Okay, so I saw all these terms in the PPT, in the lecture PPT that was shared along with the lecture. Okay, with that, we'll go to the next question. Similar kind of a question, very easy question. I think we should go about this faster. I'll just give a couple of minutes to get answer for this. Right. It's it's simply straight. Piezoelectric detects frequency, electrochemical reactions detects the redox reactions that happen. Potentiometric, again, maybe that could be a bit misleading. So it measures the electromotive force. Okay, so where this charge distribution and charge of the system comes into play. And photometric detects the light intensity, as we have discussed enough in the previous questions. So with that, I'll go to the next question. Here, yeah, calorimetric detects heat, optometric detects light. So just with the term, you can just find out. Okay, so this is the next question. Okay, so before getting into this question, can someone define epidemiology? As we discussed about census, we'll also go about seeing epidemiology in a quick review. Okay, we have one response. We'll see what others think. Cause of a disease, okay. Okay, epidemiology involves all of this. Okay, epidemiology is basically study of some disease, but what exactly we study is what are the determinants of this disease or the health state, the adverse state that is called. What will be the determinants of it? What is the distribution of it among the population? And with this two in mind, like what is the determinant of the disease and how it is distributed, we can have something like how to go about controlling the disease. Okay, so that is the whole idea of epidemiology. So our goal is to have the information necessary for taking steps that prevent, control, or maybe to take steps when something has occurred. So for all of this, what information do we need? We need the determinants of a particular health condition and the distribution of the health condition. So if we have some details about it, we can go about working on mitigating it. So that's the entire idea of epidemiology. And as you have rightly mentioned, genomics helps study new and re-emerging pathogens by identify the genetic determinants of the pathogen virulence, because one important aspect of epidemiology is to know the determinant of the disease. Okay, so here genetic determinants means what is the particular gene with which we can tell we have this particular disease or not. Okay, in case of pathogens, all pathogens work based on their genes only. Some genes are responsible for production of some protein or some kind of metabolism which is not favorable to us, and hence we term them as pathogens to us. So that genetic determinants we can study, and by assisting in the development of new vaccines and therapies. So with genomics, we can develop new vaccines. As we all know, vaccines are attenuated or they are inactive. Okay, so we have to know what inactive part of the microorganism can act as vaccine. So genomic study again helps in this, and in therapeutic purposes also we use this. it also assists in tracking the evolution and transmission of pathogens. So, as I told you, we need to know the determinants and we need to know the distribution among the population to term the study as epidemiology, right? So, in that case, we have to know, we can figure out what is the evolution of the pathogen and how it's being transmitted. So, we have a better idea about the distribution here, okay? And then by noting down the ecological factors that contribute to the pathogen emergence. Uh, all maybe A and B together work for something called that part of determinant thing in the definition of epidemiology, and the option C and D work for uh, the distribution point of view. So all this together gives us a whole idea of epidemiology, and we can go about prevention and control of disease. Okay, I guess the question and the options are clear. So as you have rightly mentioned, it's all of the above. So yes, now you can define all these three terms, sorry, four terms. I'll just wait for one more minute and then we'll discuss. These terms are important in terms of epidemiological study. Okay, we'll discuss. So, endemic is a condition that occurs in a specific geographic area or a population over a prolonged period of time. So, these diseases are common in that geographical location because they occur only in that area and they are prolonged. Any atmospheric condition or environmental condition can favor this. Um, and mostly these diseases will be infectious. So, endemic, pandemic, epidemic, maybe at least in this course we are concerned about those are infectious because we studied microbiology. So the condition in the case of endemic, it's very specific that it per- pertains to a specific geographic location and or a population for a prolonged period. Okay. Similarly, a epidemic, an infectious disease that rapidly occurs through a population. Okay. Pandemic disease that spreads in multiple geographical areas and globally. I think with pandemic, all of us are pretty sure what a pandemic is. We have been through COVID-19. We have witnessed how pandemic can change people's life as well as the global scenario. So multiple geographic area and globally um, is a pandemic. And in case of outbreak is something that uh, spreads in a specific geographic area. It's limited to the geographic area and the scope of it is temporal. Okay, so in that case it's just a major outbreak, maybe because of some floods, rains, we can see outbreak of some disease in some uh, parts of the world or parts of the country. So that we term as outbreak. I think we have received two answers and they are right. So thanks for responding for such a long question. I guess you are clear with the terms. I think we have studied about this in earlier term lectures also. Okay, that's a very specific and a simple question. So, abdominal pain, severe diarrhea, and dehydration are a symptom of cholera. Questions like this may be asked in the exam, may not be. So, we have only one response set. I'll just wait for a few more minutes. Okay, so the answer is A. See, water distribution system in the sense it's the network or the line through which we distribute water to each of the houses or the buildings in an area. So in that case, it provides a conduit for water to reach all places. Okay, whereas in the option B, what we are telling it's adding chemicals to water. No, it is not adding any chemicals to water. The water distribution system is not going to add any chemical. It's not going to create stagnant zone. It's a distribution system. So what it does is it just transports water. So it does not create any stagnant water reservoirs, and also they're given that destroy vibrio cholerae. So if it is going to destroy vibrio cholerae, why will it spread? And the last one is reducing the flow of water that uh, encourages the growth of Vibrio cholerae. No, it doesn't reduce. There is no such limits where the water flow does this. Okay, maybe relatively we can have that when the water flow is so fast it gets washed away. But ideally, why will we use a very fast water distribution system in a residential commercial places, right? So all those last three doesn't make any sense in the sense that they're asking about water distribution systems. So option A is the answer here. Here is the next question. Okay, I guess we can go with discussing about the answer for this. So, as we have studied previously, we saw about selectivity and all. So, now we'll see about this question where statement A tells high sensitivity is a preferred characteristics of a biosensor as it allows for detecting low concentration of the target substance or organism in a sample. So, high sensitivity means sensitivity generally refers to the detection limit, means how small of the analyte can it detect. When we develop any method, it's not like if I develop a method, for example, to detect starch, it's not true that even if I put maybe zero point 0.1 nanogram of starch in my solution and I'm going to give it to that sensor, it can detect that. So it basically has a range in which it can detect, below which we will get wrong results using that methodology or that sensor. Okay, so that is known as sensitivity. So sensitivity means what is the lower limit that can be generally detected. So here what they are telling is high sensitivity, which means lower and lower the concentration of the analyte that it can detect. That's what is mentioned, right? For detecting low concentration is preferred. So that is true. Okay, sensitivity, I'll repeat, sensitivity is something where you detect the lower detection level or the lowest concentration that can be detected. Higher the sensitivity, you can detect very low concentration also. For example, generally if I can detect 1 milligram, if I'm refining the sensitivity by some process modification, maybe there is chance of detecting even 1 microgram of that analyte. So that's a very important factor when we are designing a methodology, detection methodology or a sensor. Okay, and statement B tells biosensors are designed to be highly specific to detect the target substance or organism in the presence of other substances or organisms in the sample. So this is what we saw in that question. One of the options read about interference, and the other options read about this selectivity. So highly specific to detect the target, in the sense that is known as our selectivity. Okay, first statement directly talks about sensitivity. In the second statement we have the term specific, so it talks about the selectivity and the interference. Okay, in even in that case, statement B uh, holds true for all the reasons that we have discussed in the previous question that we discussed in similar pattern. So here, as you two people have rightly answered, both the statements A and B are true. Okay, so here the answer is both the statements are true, but the point here is you have to understand why they are true. I hope it's clear. If you want me to repeat, let me know in the chat box. For now, I'll just post the next question. A we have C. Okay, so we have A, C in the answer. The answer is A. See, we have different things in a biosensor, right? So the analyte is something that we want to detect, that we want to know about. So that analyte generally binds to a specific biomolecule or some bioelement. Okay, so that bioelement provides a specific binding site for the target substance or organism. So the target substance or organism is our analyte, and that binds to a specific bioelement. And this interaction creates some kind of a change. For example, we have many detection techniques as we have seen, like heat, light, piezoelectricity, other forms, many other forms, calorimetric, optometric, so all of that, potentiometric, all of this are detection mechanisms. So the analyte binds to a bioelement and the interaction brings about changes in any of this. And that interaction signal is converted into an electrical signal with the help of a transducer, and when it is, in case it needed to be amplified to show to us, right? So it's a very minute thing that's happening in the sensor, and to get a readable output there is necessary for this amplification, and transducer may involve in the amplification, but the role of transducer is not amplifying, and neither it generates the biological signal. So the biological signal generation is done by the interaction between the bioelement and the analyte, and the generated signal is converted into an electrical signal, and further if needed it gets amplified, and then we get a readable output. Okay, so that is how a biosensor works. I hope it's clear now. So transducer is generally something that converts a biological signal into an electrical signal so that we get a readable output. Easy question again. Okay, so we have multiple responses, and yeah, Rachna, please note that all of them are examples of biosensors, including C. So another question. We are just have a couple of more questions, both specific questions to some technique. Okay. Okay, so let's see the answer for this question. It's surface plasmon resonance in ion selective electrode. So first of all, you need not be very well versed in knowing about all the options. Uh, but the thing here is they have given that it is an optical sensor. So and it uses a label-free technique that detects change in refractive index on the surface of thin metal film. So here what happens is, I think in the other case you will be very sure about its ion selective electrode because it's a potentiometric biosensor. So in the case of potentiometric biosensor, you are not going to use quartz crystal microbalance. It's piezoelectric. It's not potentiometric. So that part of the question is a bit comparatively easy. So now you have to fix upon whether it's ELISA or SPR. So what happens is, even if you don't know anything about this, you can see enzyme linked immunosorbent assay. The term completely deals with a biological detection thing. Okay, it's like it uses enzyme, it uses immunosorbents. Like you can split the word immunology and sorption. So this nowhere can comparatively have a relation with refractive index. So the key term here is the refractive index. We know this plasmon resonance. Okay, so this resonance term is kind of a physical term. Physics deals with some physical interaction. So maybe even if you don't know such words, and even if questions like this come, what you can just link it and try answering it. Okay, so in the purview of exam I'm telling this, and if you're going to generally apply it to any exams, just be cautious. of negative markings. Okay, because few of you have already asked me about gate exams, other exams also. So this session is purely dedicated for the NPTEL course and the exams. General caution to relate and to answer based on the nature and the type of exam that you are attending. Okay, so with that here, uh, surface plasmon resonance usually detects a change in refractive index. I think now you know and now you will keep it in mind. If you get something out of the box, just try connecting, because you have only two options. It should be either SPR or ELISA. ELISA sounds completely biological, so in that case you can link this and you can give the answer here. Okay, so and to talk about ELISA, in ELISA, generally what we use is an antigen antibody interaction. We just quote some antibody and the antigen. Okay, I guess if you know about antigen and antibody, antigen is a foreign particle. Okay, that when we are diseased, the foreign particle that causes is known as an antigen, and the antibody is the protein that is produced to counteract the effect of antigen by our body. So antibody is produced by the human body, whereas the antigen is the foreign particle. So with that in mind, what we can do is this antibody we can coat it on some surface, and the antibody is specific. Okay, so the specificity is there. So with that, when you are going to put the antigen solution into that antibody that you have immobilized, there can be interaction, there can be binding. So this binding can be detected using change in they can many detections that can be used for ELISA. So with that, you get to know, okay, whatever I have added is this particular antigen which is responsible for this disease. So in a very broad sense, that's the meaning of ELISA. So if you are going to work on it more, you can just go and have a read about all these techniques. Here is the last question. Very simple, yes. So AU nanoparticle sensor uses gold nanoparticle to amplify the Raman signal. Okay, with that we come to the end of today's session. If you have any doubts, you can post in the chat box, or if you have and yeah, as I told in the starting of the session, the next session will be the last week session of this particular 12-week course on applied environmental microbiology. So if you have anything specific to be discussed in that session, you can just put it in the chat box, which I'll note down and make sure that I do it in the next session. Also, one more thing, in next week's session, uh, do you want me to discuss only the week 12 topic, or to give an overview of all the 12 weeks that we have covered so far? Please let know that also in the chat box. I'll prepare accordingly. Okay, we have one person telling its overview. I think there are around six people in the meet. So I'll just go with the majority. So kindly post in what you need. You can also type something like 1 hour of week 12 thing. We have a 2 hour session anyways. One person, okay. One plus one, okay. Overview, okay. I think we can just have one more response and then I'll just sort it out. Okay, no one wants a pure week 12 course content session, so I'll just go with giving a 1+1 session where typically I'll try to minimize the week 12 content a bit and then give a larger overview of all of it. I'll also bring some questions that helps you brush up all the topics that we have discussed so far. If you have any particular topic that needs to be discussed in week 12, because it's a very vast portion to be discussed next week. So if you have any particular suggestion which you feel like it should not be missed out, you can also let know in the chat box. Um, Otherwise, like, yeah, thank you for joining in for the session. uh, Thank you for being consistent in attending the session. It's just gives me a good boost to prepare the questions and to interact with all of you, despite of changing some changes or some um, replacements in the session. You still are consistent in attending the sessions, which is the main drive. Thank you all for that, and hope to see you in the last session next week also. Please do join. For now, wishing you a good week ahead. Thanks for joining.